as I've told you before, the same things. DNA doesn't have in the normal course any duplicate. There are about 35 genes in human bodies and 100 million nucleotides in one DNA molecule. And the DNAs they make, they can be either mitochondrial, they can be found either in the mitochondria of a cell or in the ribosomes. It is, as I told you, I have already told you these, that is, it has sugar, it has phosphate, it has bases. The bases are four: ATGC, adenine, thymine, guanine, and cytosine. And they pair in a particular sequence. They don't pair as and how it likes. So, they pair A pairs with T and G with C. If they pair in a wrong sequence, then there won't be any match. And if they pair, how the DNA analysis is made, from that you will understand there are portions, definite portions of there known as codon. That is made up of a few base pairs to make a small, short portion, and that is a codon. And in the DNA sequence, if one part has a sequence which doesn't match with the other part, other strand, then that is known as miscodon or nonsense codon. This importance of DNA sequence has that one strand is based with its paired nucleus. That is, if there is ATGC, ACTAA, CCGAC, you can certainly say what the other strand of the DNA molecule will have. So that is, from this you can say the sequence of the other strand will be, what will it be? The other strand will be TA, GC, TC, TG, AT, TGG, ATG. Just of the binding pairs. There will be the binding pairs. Now, if this pair differs, then there is no matching between the two. So that DNA which you have found from the sample of the suspect, it doesn't match. If it differs in at least 50% of the cells, of the strands, then that DNA is not a match. And if it doesn't differ, if it has the analogous pairing bases in the other strand, then the sample of the suspect which have been found to match this will, you can say that this in suspect has the same as that of the sample which has been sent. That means what you do from the crime scene, if you just collect the DNA sample and you send it to the laboratory, the forensic science laboratory, for sampling, genome finding, characteristic finding, karyotyping, that is chromosomal typing. So if you do that, there are steps to do that, and if you do that and you find that you have to take the control from the suspect. Taking the control from the suspect and allowing both the things, the samples and the suspect sample, both undergoing the process of DNA analysis, you find that the the sample and the suspect sample, that is a control, they match. Then that person can be said that he is guilty. And if they don't match, then you can acquit that person from having committing the offense, the crime. So as I told you, if the sequence is not correctly made, then a portion of that DNA is known as nonsense codon, and that leads to the mutations which we have nowadays, usually in influenza virus or in the COVID viruses. What we are finding, the mutations we are finding. At first we are finding some DNA sequence in the COVID, And later on that is changing to another DNA sequence. Like in influenza, you can have two types of influenza virus, H and N, H1 and N1, or H2 and N2. There are 5 paired things, types of it, H1, 2, 3, 4, 5, N1, 2, 3, 4, 5, and they can pair amongst each other in innumerable combinations. So might be what influenza you are having now, you're suffering from now, say you're suffering from H1N1 virus, but later on you find that the medicines which you have given for that particular type of influenza is no longer acting because that has already undergone mutation due to perhaps exposure to medicines repeatedly, same medicine repeatedly, or due to environmental changes, or due to reinfection by some other person's karyotype virus, and there can be recombinant DNA. That means it is matching with one with the other. Both together they are producing a mixture of the two. And this can lead to that the next time when you are treating that patient, this patient is not responding because the gene has already mutated. Similarly in COVID also, the vaccine we used to get at the first time, now that vaccine is not responding on us because it has undergone mutation. So the importance of this base pair, that is one strand based on the sequence of the other, you can predict. It is responsible for replication and transcription, and it can repair, DNA damage can be repaired. About 99.9% of all the human beings have the same sort specific sequence. Only 0.1% of any individual is unique for that person. They have a specific codon sequence. And this, that means each person has 99.9% same strand, but only 1.1% is different from the other man. So no two individual has the same type of DNA pattern, sequence of DNA pattern. From where you find the DNA? DNA can be collected from all these samples, that is blood, semen, skin, cells, tissues, organs, muscles, brain cells, bones, teeth, hair, saliva, mucus, perspiration, that is sweat, fingernails. If someone is accused of a rape, a sexual assault, then his or his fingernails might have a skin underneath the nails. From that, on scraping the nails, you can take that sample and put it into the procedure of DNA analysis, and you'll find that this person had some linking with the victim. So fingernails is important in that case. Amongst all these, what we collect usually in a living subject is blood, semen, then hair, saliva, fingernails. These are main ones which we keep. The identity of the individual cannot be found, unknown bodies. You can usually take bone marrow, teeth, then a long bone with the tissues attached to it, and we tell them to preserve it without any preservative, but only on dry. Just dry it and put it for further DNA processing. So what is the use of DNA sequencing, of finding out the DNA profile in our discipline, that is forensics? To identify potential suspects, to exonerate persons wrongly accused of crime, to identify crime and criminal investigations, identify catastrophe and natural disaster victims, identify persons who are otherwise difficult to establish, that is decomposed, completely burnt, skeletonized or mutilated people. To solve any dispute of identification, establish paternity and other relationship, detect mutated genes, genome mapping and genetic disease analysis. The last one is important for certain diseases which have yet to be which has to undergo several research processes like multiple myelinosis, multiple sclerosis, multiple myeloma. These are the things where the genome typing has to undergo. But till date there is no cure for all these. Parkinsonism, Alzheimer's disease, they have no cure. They are under genetic engineering processes. Now in case of paternity dispute, that is very important, and you take the blood, or usually blood is a sample with CSFL or FSSL. It is done by Central Forensic Science Laboratory usually. They take it and there's a process of that, and they try to match it with the suspected baby who the mother tries to prove that he is the biological father of that child. But there is no such test called maternity detection. Why? Because each baby, there are two types of chromosomes. One autosomal, the other one is sexual, that is sex chromosomes. Sex chromosomes has, in case of a female, XX, this formula, XX, and in case of male it is XY. So when these two genes, they come into combination, you can have two child of XX, XY, that means two each, XX, XY. Again, if you break it down, then it will be XX, XY. So Y chromosome is more important. Hence, paternity detection is more important than maternity detection, because the father, the male person, also has X chromosome. But Y chromosome is only found in male person. So, if you can find, that is easier. That is easier to detect and easier to find out whether this is he is a biological father of that child or not. But someone has questioned also, why not the maternity detection also? Because of the fact that extramarital affairs also occur, and she can also go in for, after living with her own husband, can go in for extramarital affairs, and there the mother can be at fault. But still that is not in vogue. Then in cases of decomposed, completely burned, skeletonized, mutilated people, where you have nothing else but only the bones can be taken, because post-mortem blood has no such value in DNA examination as fresh blood. It has value but not much importance. So the bones here act as the sample, as the specimens. Tooth can act, hair can act, if not completely singed in burnt cases. If it is first degree, completely total burnt, first, second degree, then fresh burns, you will get a charred body with most of the organs burned. So in that case, hair will also get singed, and the DNA stays not at the end portion but at the bulb, that is the root portion of the hair. So DNA analysis has been established in 1980 by Alec Jeffreys, an English geneticist, and forensic scientists can 13 DNA regions or loci and create a DNA profile for that person, which is known as DNA fingerprinting. Taking apart from all the other fingerprints to establish identity, as we know, that is dactylography, iris print, footprint, handprint, poroscopy, everything, the most accurate one is DNA fingerprinting. In criminal cases, samples are collected from the crime scene and from one or more suspects, extracting the DNA and analyzing it for a set of specific DNA markers. Markers are found in the DNA by making small pieces of DNA probes that will seek out and bind to a complementary DNA sequence in the sample. More probes used, greater the odds For a unique pattern definite match, at least 4 to 6 probes are recommended. Now this is what, that is, if you collect a sample from the crime scene and then send it to the lab, what they do first, this collection is also very important. That is, they have to collect in a specific method and send to the laboratory as quick as possible in the properly packed, sealed, labeled package. Otherwise, the chain of custody cannot be maintained. This you know, I don't have to go into that. To maintain the chain of custody and be acceptable to the court, you have to document it that this was the sample which you have sent. After reaching the laboratory, they go put the DNA into 3 sequential patterns. 1, extraction. 2nd, fragmentation. 3rd, replication. And then putting them into the machines to find out the DNA nature. This I already told you, whether he is a suspect or not a suspect. And the portions must be large enough to exclude any fallacy or to give it more precision and accuracy. To show there was no random bias. That is, it has been taken genuinely from that particular specific person and has been sent for DNA analysis. CODIS is a system